في هذا diagrams will be explained about the degradation of the RBC, the aged erythrocyte. After 120 days, the RBC is degradation in the spleen to give the hemoglobin converted to give the globin proteins and non-protein. The protein is globin, and the globin may be used to synthesis hemoglobin in the body, or maybe break down to give the several amino acid. This amino acid also used to synthesis of globin in other time. And the heme part of the heme part is this structure represents the heme part of the hemoglobins. The hemoglobin by the hemooxygenase enzyme is led to breakdowns of the methylene bridge between the A and B. This viral molecules is called the A, and this B. The hemooxygenase break down this bridge between two viral molecules or ring, and liberating or to give the other compound is called the biliverdin. By presence of the oxygen and NADPH to give the NADB plus, and release of the ferrous that's present in this structure. Inside the heme is present other ferrous. The ferrous هو عبارة عن حديد يحتوي على عدد أكسيد ذات 2. After release of the ferrous outside the ring, converted to the ferric forms. This reaction occurs by the hemioxygenase and release of the CO, monocarbon, and liberating the biliverdins. Biliverdins is colorless compound, and the biliverdins is reductase, or reducing the biliverdins by the biliverdin reductase enzyme, and present the NADPH with H plus, and converted to bilirubin by reduction of this two carbon atoms, and added the hydrogen to convert it to methylene and give bilirubin. The bilirubin يختلف عن البليفردين بمجرد انه في هذا الموقع يتم اضافه ذرتين هيدروجين لهذه العصره، يتحول من بليفردين الى bilirubin. The bilirubin طبعا these reactions occur in the spleen، يعني هذه كل التفاعلات اللي نشرحها كلها بالسبلين. The bilirubin with not present of the double bond, and the bilirubin released from the spleen to blood, as the bilirubin are insoluble in water, therefore transport from the spleen to the liver after conjugated with bilirubin with albumin as carrier, and binding with non-covalently but by the van der Waals, and formation the bilirubin albumin complex. After release or after reaches this complex to the livers, the bilirubin is released to the hepatocyte and binding with ligandin proteins inside the hepatocyte, and conjugated of bilirubin with glucuronate that obtained from the two molecules of the uracil diphosphate glucuronate, and the presence of enzyme يسمى bilirubin glucuronyl transferase enzyme responsible for binding of bilirubin with glucuronate to give the molecules is called the bilirubin diglucuronate. إذن جزيئتين من مركب الجلوكورونيك أسيد اللي انحصل من مركب ال two molecules of the two uracil diphosphate glucuronate يعطينا تو موليكيولز ترتبط مع البيليروبين يسمى بالبيليروبين داي كلوكورونايت اللي هو عبارة عن كلوكورونيك أسيد binding with bilirubin into this position. This present is طبعا هذا يسمى بالconjugated، أما هذا المركب يسمى بالinconjugated أو الانdirect، أما هذا يسمى بالconjugated أو الdirect bilirubin. After that, the microbial enzymes intestine. After this compound formation in the liver, it released by the bile duct to bile duct, and when this compound طبعا reaches to the GIT by the specific bacteria, some enzyme is converted of the bilirubin diglucuronate, release of bilirubin and converted to give the yellow compound. This compound you converted to by the bacteria converted to urobilinogen. Urobilinogen may be released by the kidney and converted to طبعا this colorless converted to urobilin compound in the blood and released outside to urine and excreted by the kidney. But apart from this compound converted to the stercoline, corbilin, converted to this compound give the feces give the brown colors. This scheme explains the reactions or converted of the bilirubin from degradation in spleen and to release outside by the urine or the feces or stool. Degradation of the hematobile pigment color used in structure represent the change in the specific reaction only.